welcome to the HPL seminar. Thank you for joining us today. We have with us Jordan King, who is a PhD student in the Department of Biomedical Engineering and is doing his PhD with a specialization in wearable technology under the supervision of Dr. Ryan Peters. Jordan started his research journey as an undergraduate student in the honors program in kinesiology here at the University of Calgary in 2017. He finished his undergrad in 2022 and went on to start a master's under the supervision of Dr. Ryan Peters again here at the University of Calgary. He then transferred into the PhD program a year later and is now studying the electrical vestibular stimulation applications in balance, gait, and sensory conflict. Outside the lab, Jordan has been a competitive gymnast for 14 years and coached for 10 of those years as well. Now, he spends most of his free time outside, swimming or diving. Today, he's going to be presenting his talk on electrical vestibular stimulation to improve static balance in older adults, a randomized controlled trial.
Perfect. Hi, everyone. My name is Jordan. I'm a PhD student in biomedical engineering, and today I'll be presenting electrical vestibular stimulation to help improve static balance in older adults. There we go. Okay, we're good. Okay, so today we're going to go with a little overview of some background information. Next, we'll discuss the hypothesis for this project, what we're testing. Then we'll go into the methodology, how we did this experiment, talk about our results, interpret them, and then we'll talk about some limitations and some key takeaways from this presentation. So what you're seeing right here is a pie chart of population data from Stats Canada. On here on the left, you can see the proportions of the population based on age groups. Right now, seniors make up just under 1/5 of the population, and this is supposed to increase to 1/4 of the population by 2040. If we look at the injuries by cost, by age categories, and what the most prevalent category, we see that falls are the most common type of injury that causes hospitalizations. And seniors right now make up more than half of that budget, or half that cost. So falls are a really bad thing. Falls are a really bad thing. And to understand falls, we have to understand how people can stand and how people balance. So we're going to talk about a little bit of the sensory integration for how people balance. Balance is made up of 3 main systems. We have our vision, which is what we see and how we perceive things. We have our proprioception. So throughout our body and our muscles and our joints, we have lots of sensory organs that lets us know how our body's moving in space. And it also lets me close my eyes, touch my nose, and then touch my finger together without any vision. And then most importantly, we have the vestibular system. This is what we're going to be mostly talking about today. And the vestibular system is important for balance and stabilizing your gaze. So right now, in order to balance, your nervous system will weight these 3 systems and in order to maintain balance during different tasks and different environments. So if you are going to bed and you turn off the lights and you're about to go walk to your bed just to go to sleep, you now have eliminated vision and you now have to rely more heavily on your proprioception and your vestibular inputs to keep upright. Unfortunately, these all degrade as we get older, and vision starts to get worse. We're usually around the age of 30. Vestibular deficits are marked with imbalance and dizziness. That starts around age 40. And then proprioception just slowly goes downhill as well. So as we age, we have a tendency to reweight these 3 systems more heavily on vision. So right now, older adults primarily use vision in order to keep standing upright and to make sure that they don't fall. Currently, there are interventions for vision and proprioception. Right now, if your vision's getting worse, you can go to the eye doctor, you can get some glasses. They'll help you see longer, or they'll help you see more clearly for a longer time. With proprioception, it's been well known that doing exercises to promote proprioception is very beneficial, especially for older adults. However, once we go to the vestibular system, it's very limited on what kind of treatments that are available. Right now, vestibular rehabilitation therapy is the go-to protocol for anyone with vestibular deficits. What this is exercises used to promote vision and proprioception to make up for the deficit of vestibular loss. So we're going to be focusing more on the vestibular system. To give an overview on that next. So the vestibular system. This is located bilaterally in your ear, in the inner ear. This bony labyrinth is housed within the temporal bone, and throughout it, we have 5 sensory organs on either side. We have our 3 semicircular canals, which you can just see right here, these 3 loops. These are positioned so that they're able to detect rotational acceleration in all 3 planes. We then have our otolith organs, the saccule and utricle, which detect linear accelerations. So if you're driving forward in a car, you'll be able to feel the linear acceleration moving forward, as well as if you're tilting your head forward or to the side, it's able to detect that. It's able to detect these movements through specialized sensory cells, or sensory hair cells. So just like the hearing system, which is this snail-like structure right here, the vestibular system is filled with a fluid inside and hair cells. And as you move around, the inertia of your head movement is going to cause the fluid inside of your vestibular system to move around, and that's going to deflect these hair cells. Hair cells like to move in a specific way. So if a hair cell is pushed to one way and it links that way, it gets excited and causes excitation, sending a signal to your brain. If it gets pushed the other way, you now get inhibition. So since we have our vestibular system on both sides, if I'm turning my head to one side, you're going to get excitation from one side and inhibition from the other side. So it works kind of reciprocally with each other. So after our vestibular system goes and detects all these motions, where does it go and what does it do? So the first stop is when the vestibular system sends information, it's going to travel through the vestibulocochlear nerve, the 8th cranial nerve, and it's going to enter in the brain at the brainstem. From here, it enters into the vestibular nucleus, and then from here, it gets relayed to a bunch of different areas. We have 5 main pathways that we're going to kind of go over, or I'll go over quickly. So first, we have a projection that comes up to here. This is where the oculomotor nucleus is, and this is involved with your eye movements. So the vestibular system is important for stabilizing gaze. If I'm trying to look at something in front of me and if I have to turn my head and focus on my object in front, my vestibular system is going to detect that rotation, and then it's going to send a signal up to my eyes in order to move my eyes at the same rate that I'm moving my head, so I can keep my eyes locked on one spot. We then have some pathways that travel down the brainstem to the spinal cord. We have our lateral vestibulospinal tract that goes down and controls our cervical neck muscles. And if we are being tilted or anything, our vestibular system is going to detect that, and it's going to cause a reflex to help keep your head upright. This also works with our lateral vestibulospinal tract. That goes down and controls trunk and limb muscles in order to help with postural adjustments. We then have a vestibulocortical pathway. So after it goes to the vestibular nucleus, we also get a projection that goes to the cortex for more sensory integration and perception. And we have a reflex loop that goes through the cerebellum just to help fine-tune these reflexes so they're working properly. There's another reflex that's involved with baroreceptors. So when you lay down versus standing up, your blood pressure will change. That's also detected by the vestibular system. So the point I'm trying to make here is the vestibular system has a widespread activation throughout the entire nervous system. So that leads us into electrical vestibular stimulation. This is a common technique used to examine the vestibular system, and through stimulation, we're able to activate the vestibular system itself. So we place electrodes behind someone's neck and ears. So on the Massway process, that's that hard bony spot behind your ear, and on the neck around the C4 level. So just underneath your hairline. By placing electrodes in these specific areas, we're then able to stimulate the vestibular system, the hair cells themselves, as well as the nerve. When we use higher levels of stimulation, we're able to cause eye movements and postural perturbations. So this is a common way that researchers use this in order to see how the vestibular system works. They'll get someone to stand, they'll place electrodes behind, and if the participant closes their eyes and we send a specific waveform, we can get them to lean to either the right or the left, depending on how we're sending that electrical current through. There are different ways that we can send that electrical current as well. So one way is through direct current. That's when you just blast at a specific amplitude and it stays constant for that whole time. For example, if we were doing 2 milliamps of direct current, the stimulation would go to 2 milliamps and it would hold that constant 2 milliamps the whole time. There are other waveforms as well. There are sinusoidal waveforms where the current will go up to 2 milliamps and then down to negative 2 milliamps and then slowly go back and forth. And we also have a noisy stimulus. By having Gaussian white noise applied and having filters cut off to for the amplitude, we're able to send a noisy signal, and that is less predictable by the brain. So right now, if we send direct current to the vestibular system, our brains are able to adapt to that over time. But if we send a noisy waveform, your brain isn't able to adapt to that, and it's able to constantly stimulate it the whole time. There are different ways that we can send the stimulation as well. We have higher amplitudes, or high intensity, and at higher intensities, that's where we start to evoke those reflexes with eye movements and postural perturbations. At lower levels, we still see activations of the entire vestibular pathways. However, we're not evoking these reflexes anymore. So our study will be focusing on the low intensity broadband stimulation to see if that can help improve balance. So how do we get to seeing if this can improve balance? Well, first, there were some groups that have already looked at this. They're curious to see if we shock people while they stand, can we see improvements in their balance? And they did. There was one study done by Fujimoto in 2018. And in that study, they got older adults to stand and they gave them electrical vestibular stimulation, or EVS, and they were able to see their postural sway was decreased during the stimulation, and these effects were lasting to up to 6 hours after the stimulation. So that led us to think, well, if we do this more often, could we see larger improvements to balance? And if we do this more and more, will we retain these benefits longer? That brings us to our hypothesis. So we propose that sub-threshold wideband noisy EVS can help improve postural stability and balance in older adults by driving neuroplastic changes to the vestibular system. Our prediction is subjects receiving the stimulation will show balance improvements that accumulate through the intervention, and these will be maintained through our follow-up sessions. So in order to study this, we needed to recruit participants. We recruited participants aged 50 and 50 plus, and we ended up getting a range from 50 all the way to around 95. We excluded any participants if they used a pacemaker, a hearing aid that was not removable during the electrical stimulus, as well as any neurological or musculoskeletal injuries that affect balance. We recruited participants from October of 2022 all the way to November of 2023. We originally started collecting our participants in lab, just across, or I guess right beside this room here, and it took us about 5 months in order to collect 15 participants through the 6-week intervention. So in order to mitigate this and increase our data collection, we partnered with senior homes around Calgary, and we went to the senior homes. We gave a presentation explaining what our research is, asked if anyone was involved in participating, and by doing that, we were able to over triple our data collection at a time. It was awesome. It was really good in order to get that, as well as trying to recruit older participants, especially 70 plus, to come to the University of Calgary in order to do balance testing was a huge barrier as well. So right here is a consort flow diagram showing how our participants flow through our study. We originally had about 65 people interested in participating, and we excluded 17 people. 10 because they didn't fit the inclusion criteria, and then we had 7 people that just didn't want to participate after finding out the study commitments. After that, our participants were randomized into equal groups of the stimulation group and our sham group. Our stimulation group will be receiving the stimulus, while our sham group does not. Both groups went through the intervention, and we lost some participants due to follow-ups. So we lost 7 in our stimulation group and 11 in our sham group. And then for our final analysis, we only included participants that were able to do all of our testing. So we ended up cutting 8 more participants from our final analysis. So how we did this. We had an intervention that lasted 6 weeks long. We had participants come into our lab or come in 3 times per week for a total of 18 sessions over 6 weeks. When a participant first came in during the intervention, we would place a sensor, a wearable head-mounted IMU that goes over their mastoid process on the right ear, and we ran through some balance testing and some gait testing. After that, we removed the sensor, did our electrical stimulus for 20 minutes, and then after the stimulation, we put the sensor back on their head and redid all of our balancing gait assessments. For our follow-ups, for 3-month and 6-month follow-ups, we did our balance and gait testing 2 times in a row. We did it twice because every day during the intervention, we had participants repeat those tests. So just to keep it consistent, we repeated those tests twice for our follow-ups. For our actual balance assessments, we placed the sensor behind their right ear using double-sided tape. And with the sensor, we're able to pair that to an app on our phone. From there, we're able to run through all of our balance assessments, and all of this data is uploaded immediately to the phone and then to the cloud for us to download it and then analyze it. We have 4 conditions that we'll be focusing on for this presentation, and these are our balance assessments. We had all participants were instructed to stand with their feet together, arms by their side, chin parallel to the ground, and to stay as still as possible for 1 minute during the test. We did balance assessments with older adults standing on the floor with their eyes open for 1 minute, standing on the floor with their eyes closed for 1 minute, and then we had a foam pad that the participants stood on, and we got them to repeat those. So they did 1 minute standing on the foam with their eyes open, and 1 minute standing on the foam with their eyes closed. We then did some gait assessments where we did a 25 feet walk and a timed up and go test. However, we're still in the process of analyzing that, so we're not going to be discussing any of those results today. For our electrical vestibular stimulation setup, we got electrodes placed behind the ears and neck for all of our participants. Then we had that connected to a stimulating box, and then that was connected to a laptop where we had a custom program in order to deliver the stimulus. When we got the electrodes placed on the participants, we would turn on the stimulation box that sends a little discharge at a very low amplitude. So it feels like you just get a little tap behind your ears. So we did that for all participants, just so they know that it's on. And for the participants in our stimulation group, we would press run on our program. For the participants in our sham group, we would just turn the box off, and then 20 minutes would go by. After that, we would remove the electrodes, place the sensor back on, repeat those balance tests. So this is us at one of the senior homes. This was at Fish Creek United Active Living. This was one of our first days, so you can see we had 4 participants that we were able to test all at the same time, and we had lots and lots of research assistants helping out with this project. Over here, we have pictures of another research assistant testing one of our older adults on the phone trial, and then where we did all of our walking assessments was just in the halls of the senior home. Okay, so data processing. So now that we got all of our participants to stand and we measured their balance, what do we do with all this? You can see in these graphs at the bottom, we get a time series and we plot our accelerometer data. And this shows us kind of the magnitude of how much they're swaying over the course of this 1 minute. So here on the left, we have eyes open and eyes closed. You can see that the amplitude of the signals are a little bit higher for the eyes closed, which is expected because when your eyes are closed, you start to sway a little bit more. We then took this information and we made a power calculation. It's called the vibrata power calculation. And in this, we took the magnitude of sway, times it by how much the brain weighs, times it by a time scaling factor by 0.01, just because of the frequency that the accelerometer was recording at, and we times it by gravity. This gives us a total overall energy of how much someone is swaying. The higher this value is, the more sway there is, which means that there's more postural instability. And then in order to compare across all of our subjects and our groups, we normalize all of our participants' data to their day one performance. Meaning whatever they did on day one, or they're standing, we get a power value of, let's say 1, just to keep numbers easy, then that's their value for day one. On the next day, if they get a power value of 0.8, we then divide that by 1, and that would be their day 2 score. So we do this for all subsequent days in order to look at their performance. So all of our numbers are kind of done in proportion to their day 1 performance. So right here is a plot looking at someone standing on the floor for 1 minute with their eyes closed. This is like a bird's eye view looking over top of someone as they're standing. So here on our x-axis, we have the anterior posterior acceleration, so how they're swaying going forward and backwards. And then on our x-axis, we have the medial lateral plane, so you can see how they're swaying left to right. Up here, we have our sham participant, and we have a stimulation participant down here. On their day one, this is just to show you what it looks like throughout 1 minute of standing. And you can see that in our sham participant, there's no real difference or no real, I guess, relationship looking at how they're standing. If we see the points get more condensed, then they're moving a little bit less. For our stimulation group, it's small in this particular example, but there's a reduction in how much they're swaying. Okay, so now I'm going to go into the results for each of the 4 conditions that we are testing. So in order to analyze this, we use a linear mixed model in order to look at the differences between our groups, differences between the days throughout the intervention, so the 18 days that we did, plus our 3-month follow-up and 6-month follow-up. We then did a pairwise comparisons to compare each subsequent day to their day 1 performance, just to look at if the intervention is working, when does it start working, and how long does it last. So right here, we're looking at our floor eyes open results. This is just someone standing on the floor with their eyes open. So they have their vestibular, vision, and proprioception all working in this condition. From our results here, we do not see any significant group term, so no differences between our group, no differences between the days in this condition as well. And then our interaction term, our group and day term, is looking at the rate of change throughout the study. So for all of our conditions, unfortunately, it's insignificant, but that's due to the power of our study, which I'll go into a little bit more at the end. And then when we look at our pairwise comparisons, comparing our subsequent days to their first day, there's nothing significantly different from their day 1 performance. So next, when we look at the floor eyes closed, we see a relationship start. So anything with a star above it in red shows that that day is significantly different than their first day. If there's a blue star above, that means for the sham group, that day is significantly different than their first day. For our floor eyes closed, now that we've removed vision, we have a group effect. So there's a difference between our stimulation group and our sham group. We do not have a significant day effect, so over the progression of this study, it's not significantly different from their performance, and then again, no interaction term. But if we look at the pairwise comparisons of this, we start to see that for the stimulation group, we start seeing significant differences to their first day balance performance, starting around day 5, but after day 11, it's consistent all the way up until the end of the intervention, and it's maintained on the 3-month follow-up. However, we start to lose it around the 6-month follow-up with a p-value of 0.09. Our sham group, on the other hand, doesn't have a single day that's significantly different from their baseline performance. Now, when we get participants to stand on a foam pad, we are reducing the amount of proprioceptive feedback that they're getting. By standing on there, as you push into the foam, you're not getting the same feedback as if you're standing on a firm surface. So when we're looking at the results for this, we see that we have a difference in our groups again. We have a difference for days showing that there's improvement throughout the study, and again, no significant interaction term. When we look at the pairwise comparisons with this, we see that the stimulation group again shows a earlier onset for days that are significantly different from their first day performance, and that lasted from day 12 all the way to day 18. For our sham group, we only had a couple days that were actually significantly different from the baseline performance, which was day 14 and 16, and then nothing really for their follow-ups. They do look kind of similar when you're looking at the graphs, but now that we have vision back involved, we see that that relationship, or I guess those group differences, aren't quite as strong. Now, when we remove vision and we get participants to stand on a foam pad with their eyes closed, yes, we made 90-year-olds stand on a foam pad with their eyes closed for a minute. They hated it, absolutely hated it. But we can see with the results that this is where we see our biggest effect. We have a difference between our groups, a difference of days, no significant interaction term. But when we look at our pairwise comparisons, we see for our stimulation group, their performance starts becoming significantly different after the third day. That's maintained throughout the rest of the study and maintained at our follow-ups for our 3-month follow-up and 6-month follow-up. If we look at the blue line and our sham group, we see that they do show an improvement with their balance. However, it's not as drastic as their stimulation group. We also see that for our 3-month and 6-month follow-up, these balance changes are not retained. So since we normalize all of the data, we don't get to really see how our participants looked for their baseline. So we were interested to see if we look at our participants' baseline performance, their raw value from day 1, and we look at how much they improved over the study to see if there was a relationship with that. So here on the x-axis, we have our raw data for their sway power, and on the y-axis right here, we have the differences in sway power. So what we did is we took their sway power from day 18, subtracted day 1 from it, and that gives us the difference in their performance. It's a little bit confusing, but a negative value in this means an improvement in balance, just because more sway equals higher or more instability. Same with this, less is better. So what is interesting is that our stimulation group is significant, or the worse you are on your day 1 performance can predict how well you're going to do or how much you can improve in your balance. So essentially, the worse your balance is to start off with, the more improvements there are to have. Conversely, our sham group doesn't have this relationship, and it's also not significant for them. Okay, so now that we've looked into this, saw some results, we're going to talk about some mechanisms of what we think is happening. So we're going to talk about peripheral mechanisms and central mechanisms. This first one, a little controversial, is hair cell regeneration. So it's very limited. There is evidence of immature hair cells in the epithelium of older adults age 60 plus, and there could be potential for activation or differentiation of these immature hair cells into mature hair cells. We could also be seeing an improvement between the synapse of the hair cell and the eighth cranial nerve itself. And it could also be a increase in conduction along the eighth cranial nerve too. It could be remyelination. It could just be the nerve is working better or becoming a little bit larger for better transmission. And then when we look at central mechanisms, we see that there's potentially neuroplastic changes happening, especially within the vestibular nucleus and those associated pathways. Animal studies done on mice have shown that there is that vestibular stimulation induces long-term potentiation in the vestibular nuclei, which is essentially making the synapses more efficient, so better signaling. So some of the clinical significance of this project is it's safe to use. We had no adverse effects to report for this study. We were measuring, or we were looking at headaches and reporting those throughout the entire study. The simulation did not cause any headaches or migraines in our simulation group. And this has been proven to be an easy to use, portable, low cost, and minimally invasive intervention that's specifically targeted at vestibular therapy. This could be integrated with other therapies such as vestibular rehabilitation therapy for, I guess, a more holistic approach to or balance improvements. As well, the way that this is set up, this can be integrated into a wearable device very easily, which is exactly what we were doing. So we were working with a company, Nersantis, in order to help develop a wearable device that can provide this electrical stimulus and have a way to measure people's balance. So right here is what the medical device actually looks like. We have, I think, 5 devices that are now made. We have one that's in our lab that has been modified a little bit so we can change the waveform for different purposes. And throughout the study, our participants were helping out with the design with it and with prototypes. So we had different collars that they would try on. They would see which one felt more comfortable. And based off their input, we were able to finalize on this product. And then with that as well, we have a fiber added sensor or the exact same IMU that we were using to do all of our balance testing. So this could all be easily incorporated into an app where you can do remote testing and provide the therapy. With our partnership with Nersantis, we are starting to run larger scale studies as well. So this is just a picture taken from a couple weeks ago down in the States. They have started doing larger studies using the actual device now for trying to get FDA and Health Canada approval so that we can get this out there. So limitations of this study. We did not do any a priori power calculation before. This was one of its first studies, or one of the first kinds of this study that has been done. It's the first time we've looked at the cumulative effect of electrical vestibular stimulation over an intervention. So we didn't have any data to work off of prior. So with this, we don't have enough power statistically to see our interaction term with a group in day. However, larger scale studies, especially having hundreds of people, will help start to see a more clear relationship with these. We used a single blind study design. So that means the researchers knew which participants were getting the stimulation and which ones weren't. We still have the same protocol, and the stimulus is subthreshold. So some participants weren't able to feel it at all. Some were able to feel like a slight little tap behind their ear, but we were hoping on the subtlety of the stimulus in order to be enough to have an effective blinding. And then in order to see if there was any bias with their blinding, we got participants to guess what group they were in after the study. And when we compare that to just random guessing, there's no significant difference for both our stimulation group and our sham group. And then we haven't analyzed our gait data yet, so we can't draw conclusions on dynamic balance. So key takeaways, there is a significant and sustained balance improvement in posture control, particularly in our challenging environments or when vision is limited. Individuals with worse initial balance show the most improvement throughout the study, and the portability and easy to use nature of EVS makes it a promising vestibular therapy that can be integrated into retirement homes and clinics. Thank you very much. And I'd also like to say thank you to the 13 research assistants that helped out over the past year and a half to do this data collection, as well as the Hotchkiss Brain Institute for funding me, and the senior homes that we were partnered with in order to do all this. And of course, Nersantis.
Would like to start us off. Venus.
Thanks, presentation. I'm wondering about your sway power.
Yes.
Measurement or calculation.
Yeah.
How sensitive or changes?
Very sensitive.
For the sham, the values were from 2 to 12, 14, and there was no changes or nothing like there was the sham 2.
Yeah, it is very sensitive. Getting someone to just stand normally, we'll see, pardon? The accelerometer is placed on the massoid process behind the right ear. So when participants are standing, they should be standing pretty rigidly, and then we should see kind of a magnified effect from the feet up for their sway if they are standing rigidly. It is sensitive. If someone is talking with a sensor on their head, we can see that in the actual data as well. If someone sneezes or if they turn their head, that's picked up with very large artifacts.
So how can you explain the variations and or the changes in the child movement?
Changes in the shaft group, oh my goodness, I went way too far. My bad, sorry. Two. Okay, so when looking at these graphs here, we see lots of jumps in there. We also, if you're looking at the scale, this goes from 0.75 to 1.25, and same with these ones too. We do have quite a bit of variance within it. I can't really speak to why performance would be getting worse all too much. Like for here, for example, we see almost for everybody on the third day for the sham group has slightly worse balance than after doing it for a couple days. So that doesn't entirely make sense, but the differences between like 1 and I guess 0.1 would be like a 10% increase in sway. So not a huge difference, but it's still quite variable.
Try to compare the correlation of this device with something like the sway measurements you get from the force platform?
Yes, we have. So these sensors have been validated against force plates, and a study done in 2021, that's when they validated it for force plate posturography as well as gait testing as well.
And so I'm not in the balance field, but I know there's lots of different metrics
So many, yes.
There's like total path of the center of pressure, and then you got maximum deviations, variations, 95% ellipses.
Yeah.
And the contact measurements, like what does this thing represent?
This represents how much someone is swaying or how much they're moving throughout a 1 minute trial.
So would that be like a total distance traveled?
Yeah, it's the power of how much they're swaying. So it's the magnitude and then it's converted into wattages. That's why we times it by the mass of the head, a time scaling factor, and gravity.
Good talk, Jordan. Can you talk about more about what do you think the mechanism is again? You had that mechanism slide, peripheral, central, but I'm curious, like centrally, in these older adults, is this causing, like, let's say it is working and it's improving their balance, is it causing their nervous system to pay more attention to the vestibular input or less attention? What do you think?
We're thinking that it would help increase the amount of, or help increase the actual central processing of these signals. So once the signals are sent from the vestibular system to the central nervous system, it'd be an increase in efficiency along these tracks. So it would be easier, or I guess, more sensitive to postural adjustments.
Yeah, it's tricky.
Yeah.
While we're waiting for other questions, I have one.
Yeah.
Do these in senior homes, do these participants also go through other interventions or training programs that they're participating in? And if so, could that affect how you're seeing results?
None of the participants that were in our study, actually we had two participants that did another study. I don't think it would affect their actual balance performance. They participated in a walking study where they walked about 50 feet total. I don't think that would like really change anything. So it wasn't a big intervention either. And not a lot of them like were doing interventions. It was usually just like something for them to do now during the day and to learn more about their balance.
So if you would currently the gait analysis, what do you have with that?
With the gait analysis, we haven't analyzed it yet, but based off of like calculations that we've done last year, just on some preliminary data, we saw some decreases in how much they're like, going medial laterally while they're walking, and how much deviation there is going up and down as well.
What was the variable?
What was the variable for that?
What did you measure exactly?
In this study, we were measuring,
What, if you would have done
the gaits.
analysis?
We would look at how fast they're walking, we would look at how much deviation there is left or right with their head. We look at how much their head's going up and down.
So the head would be X, Y, Z.
Yeah.
And then you would delta X, delta Y, delta Z.
Sorry, can you say that one more time?
So you would identify the location of the head,
Yes.
and then you would determine the deviation of that
Yeah,
location, delta X, delta Y, and delta Z.
yeah.
And then what would you do with that?
We haven't started, well, we have someone else that's starting to look at analyzing that. I haven't touched that data too much yet, so I can't really speak to that yet. What could we do when we're measuring it? Like,
Once the head is moving side to side, up and down, what would you pull out of that?
we'd be looking at, specifically the accelerometer that's looking at medial to lateral deviation, and then we would quantify that and kind of average it over, I guess, if we're looking at the 25 feet walk, we would average that over their walking time and then see how much deviation there is. Does that answer your question a little bit?
I think when you, start looking at vibrations and things like that, all of a sudden people, and you have no results or you have great results, people go, oh yeah, but like maybe you didn't have the right frequency, or maybe you didn't have the right magnitude or whatever. But I'm just wondering, are you guys thinking about that kind of stuff, or thinking about the frequencies you're delivering, the magnitudes of the vibrations you're delivering? Do you think that plays a role?
Yes.
Do you think maybe you've got kind of a lazy zone that you can play with and get the same kind of effect?
So right now, there isn't any like standardized parameters for the electrical stim. We have seen that subthreshold has been, or I guess, lower intensity stimulation has been easier to handle, and it's not causing any eye movements or postural perturbations, so it's more comfortable. And then with the wideband, or I guess more noisy, or it's higher frequency, we see that that's better for helping to like kind of improve like balance ability. Just based on previous literature, they found that high frequencies is better for improving balance acutely. So that's why we chose that.
Better than what?
Better than doing a low frequency. So where the sine wave's just going back and forth at 1 to 2 hertz. So the hertz that we were using was from 0 to 300. So quite high.
And then I missed it. Is it just kind of random noise? Random?
Yeah.
Yeah.
Random Gaussian white noise.
And then are there people that have an opinion about the kind of noise you deliver? Oh, it needs to be Brownian noise.
Fortunately, no.
Okay.
This is a huge problem in the field, right?
Yeah, there's just no standardized like protocol for this yet. It's still pretty novel, so hopefully we can contribute to the field with this and at least try to set some sort of baseline for an intervention for people to work off of and fine tune.
Venus, and then Reza. I think you talked about the mechanism a little bit, about the hair cell driven stimulation. So do you think then, the cilia, the cell, or the hair, they become stiff also at some point? They change their stiffness, their mechanical properties?
Yeah, so as you age, the hair cells just stop becoming as reactive or I guess as sensitive as they were before. So
So why?
it's just damage to the hair cells, and that causes them not to activate anymore. And that just happens naturally with aging. I don't know what exactly is happening to the hair cell that causes it for less signal detection, but yeah, stiffness could definitely be it. Yeah.
Okay, thanks for your talk. So clearly you explain it. I was wondering if the product that you showed at the end was exclusively based on your study or other studies also?
That was primarily this study. Yeah, so this is kind of new within the last, I guess, couple of years of can we deliver this as a therapeutic intervention? And if so, could we make a device out of it and make a wearable device out of it? So it's all started about 3 years ago in order to get this process going. And we now have about 5 products made, and they're all made and manufactured in Toronto.
Was that your primary intention in this project?
The primary intention is just to see the science behind that. Does stimulating the vestibular system actually improve balance, or is it just some sort of placebo? Like, what's happening? So that's why we did this study for, I guess, more to understand it better.
Okay. And in between, you kind of came up with the idea that you can use this study to kind of develop this product.
Yeah. So right now the company is doing larger scale studies and hopefully getting FDA and Health Canada approval.
Okay, so Hoa has an online question, then Aaron, Benno, and Matt.
Go ahead, Robert, just go ahead.
Well, first of all, thank you for a nice, very clear presentation. I'm wondering if the stimulus group is interpreted as exercising the neural pathways directly, and any improvement in the sham group might be because of the physical acts of exercising, and so by exercising on the foam and things, they see a better response. I'm wondering if the sham group simply continued to do the processes for 3 or 6 months as physical exercise, whether they might end up with similar benefits to the stimulation group. What do you think of that?
So that's a very good question. Right now, what we kind of think with that is, if you do practice these tests over and over again, you will get better. The thing is that this kind of like vestibular rehabilitation therapy, where you're now training your vision and proprioception to kind of outweigh the vestibular system. So we're thinking that the sham group is just doing this out of learning with better proprioception, versus the stimulation group having a more enhanced vestibular function. If this was continued on for like 6 months, I think that you would see that there would be more reliance on their proprioception and vision, and not so much with the vestibular. And it would also be more delayed with their learning, or I guess with their balance improvements.
Right. You showed earlier research along those lines. Has that actually been tested?
Not that I've seen. Well, that's also why we had a sham group in this, just so that we can see if this is due to the stimulation or just due to simply learning how to do these exercises over the 6 weeks.
Right. Okay. Well, thank you for a very clear presentation.
Thank you.
Aaron?
Yeah, so, you said that when they were standing on the pad, there was less proprioceptive feedback.
Yeah.
And that led to a greater amount of sway.
Yeah.
So if you were able to increase that feedback by having some type of stimulus in the shoe, like I don't know, a pebble, would you expect to see less sway?
That's interesting. I think if there's like a pebble or something that's like kind of sharp in one spot, that would kind of make you sway a little bit more, because you're like, what is that under my foot? But if there was like vibration going on in the feet, that would, I think you would see more sensitive or less postural sway with that, given those, yeah. And that has been shown too with vibration in the soles of the feet while standing.
The variable that you measure is influenced by things from the local system and things that come from the bottom. So if you put the person, for instance, on a horse platform, then you will see that the bottom has certain oscillations
Yeah.
Which you will see or not up at the head.
Yeah,
Did you check that? Because I did in my young years, it's a measurements of human micro vibrations. I call that the vibration of the human body by standing quietly.
yeah.
And there, the dominant factor was always the heart.
So placing it around the chest level? Yes,
No, measuring the ground reaction.
ground reaction force. We did do some force plate analysis. We haven't analyzed it with the force plate to see how different that is. But when we get people to stand, we're making an assumption that they're staying rigid and they're standing as still as they can. So then anything that is being moved by their head is quantified as sway. So it might not be perfect, or it might not be exactly the same as like center of pressure traces, but we're trying to see at the head level what's going on. Because that's where the vestibular system is located, and that's what's detecting these changes as well.
Why couldn't you use a force plate?
We could use a force plate. We could do force plate posturography with this,
Not the seniors.
but in senior homes, yeah, we can't. It's harder to bring force plates to senior homes, and with a wearable accelerometer, it's like more portable and easier to test.
Now the question is what the signal is. You know, you have a signal now and you don't know exactly where it is coming from. It could be strongly influenced by the heartbeat.
It could be, yes.
So you don't know that.
I do not know that.
Do you see a regular beating pattern?
We do, yeah, we don't see a regular beating pattern throughout our raw accelerometer data.
It looked, well, it looked quite regular to me. Now, go back.
So you're talking about like this red line that's oscillating?
Looks to me that it is quite regular.
That's something that we should look into more. I don't believe it's making an effect on that, but it definitely could be. These sensors are very sensitive to any movement, so anything that is kind of going on in your body should be picked up on it.
Matt had a question and then Brett.
I was looking at that first figure that you showed, the one where, it's the first one where the eyes are closed, and you have a statistical difference.
Yes. This one.
Yeah, and, it sort of seems like there are some distinct, phases almost, like it's almost like it's definitely not linear. There seems to be like different phases of what's
Like improvements?
yeah, how the
Yeah.
curves are changing. And so I kind of have a two-part question. The first part question is, I see like you use these linear mixed effects models, so you've got your time variable as day, I guess. And
Yes.
then you've got your outcome. And so you're looking at like, your day has
We have 18
not
days.
18 levels,
Yes.
right? And, you know, it's, to me, what I kind of see is a time series. And I was wondering if you explored any other time series analyses that might help you get better resolution in what's actually happening in the data.
We haven't done that yet, but I am very curious to know a little bit more if there's better ways to analyze it.
So, I mean, I'll leave it to you to figure out, but there's another, well, I'll help you. I'm happy to like share what we've done, but I'm not saying you should or you shouldn't do it. I'm saying if you wanted to chat, if you have.
I think checking that would be a good thing.
GAMs, generalized additive mixed effects models. So there are variations. So you have a linear mixed effects model.
Yeah.
But a GAM, a generalized additive mixed effects model, allows you to fit wiggliness into your data. So you can actually do a time series.
Yeah.
And you can actually fit, essentially, you could fit like splines into it, and you could then fit your data to account for all what seems to be really interesting features.
Yeah.
And then if you haven't explored it, I wonder if that would be a neat way to do it, because it might give you more.
I was considering that too, because like with this statistical test, we're assuming that going to be improving at a linear rate, but that might not be the case. It might be
Doesn't seem
exponential.
linear.
Exactly, so I think that'd be definitely something to look into.
And so my part B question is, the fact that this is a non-linear, seems like a non-linear response over time. Does that inform any of your potential mechanisms? Like, is there one mechanism that might be more explained by a non-linear change? Meaning, like at some point in time, you already see, you see it drop.
Yeah,
Could that be explained more by neuroplasticity or by these hair cells that are regenerating? Like, does it give you any evidence either way?
I'm not too, I don't think so. That's really interesting though. I'd have to know, I guess, more about the like neurophysiology about that to see which ones would adapt in different ways. But I would assume that it would be kind of just like the neuroplasticity kind of changing that mostly. But
Yeah.
we'd have to have like animal models or different ways to look at that directly.
That's sort of what I would think.
Yeah,
for plasticity.
yeah.
Brendan had a question and then Hoa.
Oh, so I had thought of something really, you can't quantify hair growth in these things, right?
Not in this study, no.
Okay.
Yeah.
All right. I do have a question about just postural sway.
Yeah.
I realize I don't have the answer to. So you have this constant exchange between the center of gravity and the center of pressure.
Yeah.
And you have sort of some options, but you can either sway really far past and then try to sort of jerk
Yeah.
your way back.
Correction.
I understand that would be quite stupid
Yeah,
to go too far. But then I don't understand why you don't just do these really quick, little small corrections, kind of like vibration.
yeah,
Do people, so it's sort of an optimization problem. You don't sway too far, you don't sway too little,
yeah,
but you sway.
yeah.
So do people know why you don't sway small and fast?
No, I don't know why. Usually when sway does get worse though, like, it is like known that higher sway, especially to a certain point, is an indication for falling, so that's the most I can kind of dive on that one. But that is interesting, if someone just standing there moving quickly.
Well, I suspect like energetics comes into play there, but I just didn't know if that was something that people know a lot about.
Or the inertia. Right.
Yeah, yeah, that's true, the inertia of the body, and you're not able to make those corrections fast enough because it takes time to travel. So that could be a potential mechanism for that.
Not really follow up, but did they move their hands, their arms?
Arms by their side.
But you forced them, like, don't.
Yeah. Yeah, we told them to keep their arms by their side. Yeah. There's like sometimes where like their arms would be like a little bit off to the side and like not directly against their body, but we didn't have people out like this trying to balance.
Hoa had an online question.
Well, they have two questions. Question number one is, what is the relationship between the used outcome measure, for example, sway, and the risk of fall? That's first question. Second question, do you have any thought on why the intervention was less effective when tested with vision?
With vision.
Yeah.
We think that, well, first of all, more sway, and sway is, the higher your sway value is, or sway power value, it's more of an indication, or it's an indicator or biomarker, I guess, for fall risk. Those have been correlated pretty well together. And then for the second part of that question, could you repeat that again, sorry?
Do you have why the
Oh, yeah.
intervention was less effective when testing?
Yeah, so we think that it was less effective when testing vision is because older adults, as a natural strategy, will use their vision more in order to keep upright. However, and that's why we don't see as strong effects on standing on the floor with your eyes open or phone with your eyes open. But when we close our eyes, we start to see those effects a little bit more stronger, because then that's where you have to rely on your vestibular system more, and that's what we're targeting at improving.
Can I jump in on? I'm trying to figure out the more the sway, the more fall risk. Like somebody did a study where they took a whole bunch of people and had, measured quantified their sway.
Yeah.
And the ones that higher sway had more falls after.
Yeah, exactly. They also looked at people's sways, and then they did surveys on how many times they've fallen within the last year or throughout their lifetime. And then there's also strong correlations with that as well.
All right, any last questions for Jordan? For your wearable device, so is it set like to give continuous stimulation or stimulation for a week and then they stop and then, so how do you see it?
With our actual device, right now it's set up to only deliver the 20 minutes of noisy sub-threshold EBS. We had to adapt it a little bit for our ProTech that we have here, just so that we can change that waveform. But it's paired with an app, so once you are on the app, you're able to create your profile, set how many sessions that you want to do, and then it will run you through placing the sensor behind your ear, doing balanced assessments. Then when you place it on, it's 20 minutes. You can't change that. You can't increase the amplitude or decrease it. And it goes for that whole 20 minutes, and then it stops. And then it will prompt to see if you want to do additional balance testing after. So that's how that device works right now.
So how do you see it as an intervention later? Like, would you then advise people maybe to do it, I don't know, once a week, twice, 3 times a week?
Yeah, 3 times a week is.
Always?
That's what we're trying to figure out too with this, is how much do we need and how long do we need it? And then how long do these last? So we'd have to do some more studies to really tease that apart to find out what's truly optimal. But right now we're starting off with this as a baseline of 3 times a week for 6 weeks.
Right, then let's thank Jordan for his presentation.